So it's a great pleasure to introduce Marnix. Marnix is actually in Wageningen, as you can see there in Netherlands. So there is a huge time difference, and nevertheless, he chose to give the talk live rather than record it. And this shows definitely his incredible dedication and interest also to support the JGI user meeting. Very brief introduction about Marnix. I met him, I think, about 10 years ago at a GSE meeting, Genomic Standards Consortium meeting. At that time, he was a postdoc at Max Planck in Bremen. And I was told that you need to meet this incredible whiz young scientist who wants to create standards for biosynthetic gene clusters. And I said, no way, it's impossible. He's dreaming, there is no way he can do that. Senior scientists or people that have been experts for more than 10 years, 20 years in the field were not able to do that. A young scientist will be able to do that out of nowhere. And nothing made me more happy than seeing Marnix proving me wrong. In 2 years after that, 2015, he actually managed to put together an incredible, the community together, an incredible actually number of scientists in the field with a lot of different viewpoints, let's say, in the analysis of biosynthetic gene clusters. And this was really, I think, a foundation for an incredible development after that of the whole field. And Marnix actually played an incredible role in further development by then taking a position at Wageningen and then launching a series of tools that have become actually the state of the art for biosynthetic gene cluster. If you are working in the field, you definitely know anti-SMAS, which is, I think, already in its 7th version. But other resources as well, like MyBig for biosynthetic gene clusters with known function, etc. So with no further ado, I would like to introduce Marnix, and Marnix can take the way.
Thank you so much, Nikos, for this wonderful introduction. It's a real pleasure to see you at least through the screen again. It was great for me also to visit the JGI just 2 weeks ago and meet a number of you there live. Unfortunately, it wasn't possible for me to travel twice within 1 month to California, even though, of course, the Californian weather is fantastic. But I hope you're having a fantastic meeting, and I'm glad that at least in this way I have the opportunity to participate and can tell something about the research that we're doing in Wageningen and partially also in Leiden. So yeah, I think Nikos already very nicely introduced like the enormous genomic diversity that is out there, all the genomes that have been sequenced for a large part also at the JGI, and the enormous functional potential that we can find in these genomes. And in my talk, I would specifically like to highlight, if we look at these specifically also the microbial genomes, but the same could also be said for example, plant genomes. If we look in these genomes, we can also see a lot of chemical potential. So a lot of potential of these organisms to produce wonderful diversity of chemical molecules that are really of key importance in the interactions that we observe between organisms. For example, if we look at microbiome interactions, let's say in a plant microbiome, these small molecules, they really play roles in all the interactions that we see between the plants, the fungi, the bacteria and archaea. We see, for example, that fungal pathogens may use these specialized metabolites, small molecules, as ways, as for example, as toxins or pathogenicity factors to invade the plant. On the other hand, certain bacteria may then try to inhibit the growth of these fungi by producing certain antifungals or other molecules. The microbes are interacting with each other. The plants are actually modulating the microbiome by exuding cocktails of both primary and secondary metabolites. And there's all of these interactions that are going on in such a microbiome community that are driven by this chemical language of the microbiome, so to say. And these specialized metabolites, I refer to them as the chemical language of the microbiome. They really play all these sorts of roles that we could envision also in for example, a language, like you may be speaking to each other as well during the annual meeting, sometimes indeed kind of casually chatting with each other, making clear you're there, for example, with quorum sensing, or with hormones, there might be, or one party might try to influence what the other party feels or wants to do. I could imagine maybe some of that is also taking place at the meeting. I hope there is not too much aggressiveness, like attack molecules, like antibiotics or these kinds of molecules. But it's been increasingly clear as well that that antibiotics are just one of the very many types of functions and activities that these small molecules can have. For example, actually many molecules are defense molecules. For example, doxorubicin, which was known as an anti-cancer agent, is actually a phage defense molecule. Other widespread molecules are actually able to defend bacteria against abiotic stresses, such as light or oxidative stress. And as undoubtedly you'll also hear tomorrow from Diane Newman, who will go into this in more detail, molecules like phenazines, but also various metallophores, are really key for nutrient acquisition. So the bacteria use these molecules to acquire the necessary nutrients from their environment that allows them really to thrive and to compete with other members of the community. And when we look at the genomes of these bacteria, as well as fungi, and to some extent also plants, we would see that the genes that actually encode the machinery to produce these molecules are clustered physically in the same chromosomal location. So all these genes to make one molecule are really next to each other adjacently on the chromosome in what we call a biosynthetic gene cluster. And when we can then analyze computationally these biosynthetic gene clusters, and there's often a very clear relationship between the chemistry that is produced and actually the sequence, and for example, the protein domains, enzymes encoded in these biosynthetic gene clusters. You see here a number of examples. For example, the coumarin group here under rhodyrine in blue is shared with the coumarin group in simocyclinon, and in their gene clusters, actually, these genes in blue actually are the corresponding genes that encode the biosynthesis of this specific chemical substructure, and they're also shared between these two gene clusters. So by looking at these gene clusters, we can often get a sense of what kind of molecules are produced, what do they look like, and what is their chemical diversity as well. And in terms of the diversity, speaking of that diversity, this diversity really turns out to be enormous. We did, together with the group of Nadine Siemert at the University of Tübingen in Germany, we published an analysis 1 or 2 years ago where we looked at all of the published genome sequences that are at the moment available, and we did a detailed rarefaction analysis based on 2 clustering algorithms. One is Big Slice, which was developed by Satria Kutsar, who I see sitting there in the room. Hi, Satria. And the other was Clustomatic, which was a tool developed by the group of Rolf Müller at the Helmholtz Institute in Saarbrücken. So 2 independent methods that were giving very similar results, which was very encouraging to us. And we calibrated these clustering methods that are kind of grouping gene clusters across genomes to see like how diverse they are. To be as granular as the chemical classes in the database for natural product structures, chemical structures, NP-Atlas, so that in this way we would be able to estimate how many of these chemical classes are actually encoded across both publicly available genomes and genomes that we may not have sequenced yet. Compared to like how the known structures for which we know the gene cluster are linked to the underlying gene cluster diversity of these known structures. So when we do that kind of analysis, we actually concluded that the estimation that really only 3% of all of these chemical classes, their biosynthesis has actually been experimentally characterized. So yeah, I think this is quite exciting because it means that really the vast majority of the chemistry that is still out there is still up for grabs, and we can actually use all these genomic data to try to mine for novel chemistry, to try to figure out what is still hidden in all of these wonderful genomes that, for example, the JGI is producing. And here at the right, you also see that when we look at like where the novelty is, this is partially also indeed found in metagenomes, in a metagenome assembled genome. Many families of gene clusters are specific to these MAGs and only partially overlapping with genomes in RefSeq. So looking at also at more exotic locations or places where you find microbes that are difficult to cultivate, you can actually find quite a lot of exciting new biosynthetic diversity as well. So how do we detect these biosynthetic gene clusters? Nico's already mentioned the anti-smash tool, and then basically anti-smash works like this. You put in a genome sequence, and using kind of logical rules combined with identification of protein domains, it looks for combinations of protein domains that are signatures for a specific type of pathway. And these logical rules can be manually tuned, manually curated in a way that, well, as long as we keep track of literature very well, is very difficult to beat with, for example, machine learning algorithms. And they can really provide very accurate identification of known types of biosynthetic gene clusters. And this is all continuously implemented in the anti-smash software that we've been coordinating the development of together with Tilman Weber's group at the Technical University of Denmark. And well, by now we're at version 7, already working hard on version 8. So when you actually put a genome sequence through anti-smash, you see something like this, where all the genes in a certain biosynthetic gene cluster, currently the 10th gene cluster of the first chromosome in this streptomycin color genome, is being selected, is visualized, and you see here all these tabs then provide lots of other annotations based on various algorithms that people in the community have been developing to actually predict the structures of the metabolites, the functions of the underlying metabolites, the biosynthetic steps encoded in these pathways, etc. So there's a lot to then further explore for each of these gene clusters. And it's wonderful also that all of these anti-smash predictions are now also available through the JGI portal, including the new SMC portal that is being developed at the JGI. So after actually analyzing these gene clusters with anti-smash, there's actually more that you can do. For example, we've built recently a set of tools, and one of them, RightU, will come out very soon, that then also allows you to take the biosynthetic pathways that are encoded in such a gene cluster and visualize the entire pathway in detail, including all the biosynthetic steps. Wonderful work by Barbara, Friederike, and Sophie, 3 wonderful young scientists who put together these tools that you can really step by step for each enzymatic step visualize chemically what is going on. And partially this can also be predicted directly from sequence. Besides like the regular biosynthetic gene clusters, we can also detect other types of relevant metabolic gene clusters. For example, we recently released, and this was work by Victoria, a new flavor of anti-smash specific for catabolic gene clusters. So gene clusters involved in breakdown of or transformation of certain molecules in the human gut and similar anaerobic microbiome environments. And this allows you to also specifically look at these kinds of pathways, which are often also encoded in gene clusters. For example, short-chain fatty acid production, but also, for example, bile acid catabolism and many other pathways that are relevant and niche, defining really niches in the gut. And similarly, at the moment, we're developing RizoSmash, another flavor of anti-smash, which is actually aimed at identifying catabolic gene clusters for root exodus in plants. So these are actually really the key molecules often that drive microbiome recruitment in the rhizosphere. And knowing which microbes are actually able to then catabolize which of these root exudate components and identifying the corresponding gene clusters as well as their variants, we hope that this can really help us to predict microbiome recruitments by crops as well. So you can imagine that if we run these tools on all of these genomes, especially all the enormous numbers of genomes sequenced at the JGI, we cannot just look at each of these anti-smash or gut smash or RizoSmash results one by one, but we need to have also mechanisms in place to look at their larger diversity. I already mentioned briefly the big slice algorithm that Satria worked on. Nikos introduced the reference database, MI-BIG, driven by the Genomics Standards Consortium standard, minimum information on the biosynthetic gene cluster. So over the years, we've built like an ecosystem of tools where they all kind of can talk to each other and play together. Starts by identifying predicted biosynthetic gene clusters with anti-smash, then indeed in MI-BIG, which is really a community-driven resource with a lot of input from people all over the world, where we currently host almost 3,000 biosynthetic gene clusters with known products. So enormous diversity already of biosynthetic pathways that have been characterized with hard labor in the wet lab to figure out the chemical structures of their products, the functions of the enzymes involved in their pathways, etc. And then we can take these predicted gene clusters from anti-smash together with these reference known gene clusters from MI-BIG and use tools like BigScape, which is a sequence similarity networking approach, or BigSlice, which is really for large data sets, a clustering approach, to really look at gene cluster families across multiple genomes and see how these gene clusters are really related to each other and how they're related to the reference gene clusters for which we actually know their structures and functions. And then you can also do a deeper dive with tools like Corazon by Paco Barona Gomez's group, where you can then look inside such a gene cluster family and see how the gene clusters in such a family are also phylogenetically related. But you can imagine if we do this on large numbers of genomes, we may also find indeed thousands, 10,000s, or maybe even hundreds of thousands families of gene clusters. And then we're left with almost impossible task to figure out which of these should we actually work on in the lab, which of these really are driving, for example, key microbiome interactions, or which are likely to produce the next generation of new antibiotics. Because of this, I think the really maybe most important challenge for the field is really to predict the functions of these biosynthetic gene clusters. So can we predict what are these gene clusters doing? What are the functions of the molecules encoded in terms of their mechanisms of action, as well as the ecological activities that they have within a microbial community? And this we've been trying to tackle in a project called Decipher, an ERC project that is currently halfway, started late 2020, where we've followed several strategies to try to predict and then prioritize these gene clusters for function. First strategy that we've been following is looking at really at gene expression patterns. In the past, we've already published cases where we've been able to relate the gene expression of a certain gene cluster with a microbiome-associated phenotype. We published together with the Netherlands Institute of Ecology a study where we looked at disease-suppressive soils, where actually we were able to identify a single biosynthetic gene cluster out of 800 gene clusters in the metagenome that was specifically expressed and strongly associated and also highly abundant in the soils that were actually disease suppressive, so that were suppressing the specific fungal disease. And through that association, indeed, we're able to identify that this gene cluster was really crucial for that disease suppression. But what we're also looking at is more complex interactions, where maybe there's not really one direct link between one gene cluster and one very clear phenotype. So what we can also do, for example, is look at the interactions through, for example, transcriptomics or metatranscriptomics, where we look at co-expression networks within and across organisms, and look at these at the correlations of expression of these gene clusters as well as other genes, both in the same genome as well as across other genomes. So we can get an idea of the regulation of these gene clusters as well as the effects that they have on other members of the community. And for this, Zach Wright, who is a postdoc in my lab, has been developing a really nice tool called Cobane that creates these co-expression networks of myostatic gene clusters. Here in this picture, the large nodes are these BGCs, and their smaller nodes are other genes in the same genome. In this case, within one genome, the model organism Streptomyces silicolor. And you can already see that there's certain kind of cliques forming in this network, and actually we know that some of these sets of gene clusters are really also functionally related. So to get a better idea of what's really happening, what we can also do is we can look at the transcription factor binding sites in front of all these genes to really know how are they regulated. And again, this is partially also thanks to wonderful collaboration with the JGI, where we can use the amazing DAP-seq technologies that allow also profiling these kinds of binding sites in large throughput. And here, with an example of this DMDR1 regulator, which is an iron regulator, is Streptomyces silicolor. There's a specific binding site that when iron is low, the regulator will let go and the iron acquisition genes will be expressed. And when we look for that binding site across genes in our network, we see that they cluster very nicely within this area at the top right of the network. And two of these nodes, these big nodes, were actually known SADER for myostatic gene clusters, so which made a lot of sense. But on top of this, we were able to localize a third gene cluster, a new gene cluster, which even in this model organism, Streptomyces silicolor, had gone completely unnoticed. And Hannah was able to make knockouts of the genes in this gene cluster, and through metabolomics was able to show that these actually, this locus is also involved in the biosynthesis of desferrioxamine, so SADER4s that are made by Streptomyces silicolor. So it was quite exciting to me that even in such a model organism, which has been studied for like 70 years, using this co-expression network strategy, we're able to find a new metallophore gene cluster through the functional prediction using these co-expression and regulation patterns. So a second strategy that we're following, and then with this I'll try to close off in the next few minutes, is where we can also look at structural features that are associated with these myostatic gene clusters. So we can predict, like I said already at the start, from the gene cluster sequences, certain substructures that will be part of the metabolites that are produced from these gene clusters. At the same time, we have databases of natural products with known structures, and we can recognize certain substructures or structural features that are also associated with certain biological activities or mechanisms of action. And based on what we know about these known chemical structures, we can then try to predict also what these gene clusters would be encoding in terms of function as well. And again, with an example of these iron-binding metabolites, SADER4s, Zach also implemented already like an analysis module in Anti-Smash that is able to predict all of the known substructures that are associated with metal binding, well, both for iron and as well as other metals. And these can now be predicted from the gene clusters in Anti-Smash reliably. And we were able to, or he was able to show that actually when you look at these predictions, they are able to predict very well, just with a few exceptions, that indeed which of these gene clusters are encoding the production of metallophore. So there is, in this case, a very direct link between the structural features that can be predicted from the gene clusters and the functions of these metabolites. Well, of course, this will not be the case for all molecules, that there is such a direct relationship. We do think this provides precedence for also more like widespread prediction of functions through also more global associations of structural features with functions. And we're working at the moment towards machine learning approaches that may generalize this principle. And of course, yeah, when we put this all together and we can predict all of these, well, also functional features out of this large-scale biosynthetic gene cluster data, I think there's exciting prospects of combining really isolates from microbial community, the genome sequencing of isolates, as well as metagenomics, where you can also really look at, for example, differential abundance across samples with different phenotypes, and use the functional information as well as the phenotypic information from both of these to design synthetic communities where you put certain microbes together in different combinations in high throughput. And specifically in plants, this can really be done at larger scales to really phenotype them and see which combinations are leading to what kind of microbiome associated phenotypes, and then using expression analysis to figure out which biosynthetic gene clusters really underlie these phenotypes and are causal. With that, I would like to close these our exciting times. Thanks also to all the technology developed at the JGI. I would like to thank my team and everybody who did all of the actual work. They're really wonderful people, and I would like to thank all of you for your attention.
Any questions? Marnix, I have a question. Have you checked, have you compared plasmids versus genomes to see if you identify if there is a specific set of biosynthetic gene clusters on plasmids or other mobile genetic elements versus chromosomes?
That's a great question. We haven't done that specific kind of analysis. I would predict that this would also be different across different taxa. For example, actually, the first paper of my PhD was about a plasmid from Streptomyces, which had more than 20 biosynthetic gene clusters on one plasmid. It was a very big plasmid, like almost 2 megabase pairs. But there's these extreme cases where indeed you see lots of gene clusters being put together on single plasmids. On the other hand, there's of course also other taxa which do not have these very large plasmids, but sometimes you do see specifically also some of the smaller gene clusters, like ribosomally synthesized peptide biosynthetic gene clusters that really can be niche defining, that are transferred also between different strains or cross strains with these plasmids, also in, for example, the human microbiome. So yeah, I do think that definitely warrants a deeper analysis.
Okay, great. One more question.
Hey, Marnix, great talk, Kate Duncan. So in terms of the core structure and relating this to function, do you think that all metabolites have one specific function? The reason why I'm asking is, in the natural products community, it's widely debated that a molecule could act as, communication at low concentrations and then maybe defense at higher concentrations. And do you think you'll be able to unpick this with your correlations?
Yeah, that's a great question and a great point. Well, I think you're totally right that function is basically a human box that we use to put molecules in, and of course, depending on the environment, on the conditions, on the concentrations, many molecules will fit multiple boxes or will fit several boxes depending on all of these variables. So yeah, it's definitely, you always have to be very careful with using these human boxes. Still, I think they can be useful as long as we allow also putting these molecules into multiple boxes and having several different degrees of a certain activity. Yeah, and I sometimes, of course, one single methyl group may switch also the activity of a molecule. So that also makes it quite challenging. So these activity cliffs may make it quite challenging to associate structural features with activities. Yeah, that is why we're also hoping to get more standardized large-scale data by really getting measurements of thousands of purified natural products to really get an idea of how we can link these structures to functions. But it's not an easy challenge.
Okay, thank you, Marnix. Let's thank Marnix once again for staying also